Hi everyone, it's Mr. Sinti, and today I'm going to talk to you about muscle contraction. Yeah, muscle contraction is very critical. We use it all the time. I'm actually contracting my muscles right now as I speak to you and try to describe muscle contraction. And it's something kind of challenging in some ways to understand because it involves kind of a hierarchy within the muscle to understand the breakdown of muscle. And it's also kind of a molecular conversation as well. And so we're going to be talking about how proteins change shape and move past each other in order to shorten the muscle, which is ultimately what muscle contraction is. And so let's get into that conversation. So as you can see here, this is a typical calf muscle right here. This is your gastrocnemius right here, which is connected, as you may know, muscles are connected via tendons to bone. Right here, here's the Achilles tendon right here, connected to the calcaneus heel bone right there. And so as the muscle shortens overall, it's going to be pulling on the tendon, and therefore it's going to be moving the bone, and so you have overall movement as a result of that. Now, in order to get a muscle to contract, it's going to require energy. And what's fascinating about it is that most people think that energy directly comes from food, and it does, but it's more indirect. So in other words, the food that we eat is broken down by the cells of our body into a chemical called adenosine triphosphate, and that chemical is ultimately going to be interacting with the proteins in the muscle fiber or muscle cell. So let's take a look at that. This is the molecule of adenosine triphosphate. It's a sugar, ribose, connected to the adenine, which is a purine nitrogenous base, and then it has three phosphates, so thus the name adenosine triphosphate. And so this chemical is what's being made during cell respiration in order to produce this molecule. It comes from food because the energy that's burned from food produces this, but this is the currency that's going to actually interact with the protein to make the muscle contract. So that's kind of cool. So let's talk a little bit about the hierarchy of a muscle. I think it's important because if we get into a conversation too much about protein down on this level, which is where I'm going, it might be difficult to follow that. And so let's go up here on a macro level, and then we'll come down to micro. So here's your typical bone right here. Here's the epiphysis right here. Look at this. This is a tendon. This is, again, fibrous connective tissue, which is attaching the muscle to the bone right here. So a tendon. Okay? But then each muscle is surrounded by this like kind of thin membrane called fascia, again, connective tissue. And that fascia helps to make sure that there's not too much friction between muscles, because that can generate a tremendous amount of heat. And also it helps, to slide along like that between muscles. And it also is kind of like a girdle. It helps to hold the muscles together because they're under a lot of pressure when they're contracting and a lot of weight bearing. So ultimately, the muscle, as we're going to see, is a series of connective tissues surrounding each of the layers as we go down. So it's all connective tissue. So check this out. So over here in the muscle is surrounded, so the large muscle is surrounded by what's known as epi, which means on top of, epimysium or mysium, depending on how you want to pronounce it. Epimysium, let's call it that, epimysium. So this is a surrounding layer of connective tissue, epimysium. And so within that, if you were to cut a cross-section like this and slice it, this is what this is, a cross-section. If you look inside, you'll see these little chambers within. Do you see that? Sorry for touching that. So these little chambers within, if we were to pull one of those out, can you see this thing that's being pulled out right here? This is called a fascicle. And so let me come over here and highlight that. So this is a fascicle. So the muscle's composed of many fascicles. You see that? There's a fascicle, there's a fascicle. So I've just pulled one out. But what's surrounding the fascicle, what makes up the fascicle itself, is something that goes around each fascicle, is perimysium. So again, another circle of connective tissue strengthening the muscle. Now, it's not just a wrapping of connective tissue. As you can kind of see here, there's blood vessels and there's obviously nerves that are traveling here because all the cells, they need lots of oxygen, they need lots of sugar, and they need nerve impulses in order for the muscle to contract ultimately. So again, to review, it's epimysium is surrounding the whole muscle, and then each surrounding fascicle is surrounded by perimysium. So check this out. When you pull out a fascicle, and there's one fascicle right there. So this is a cross section of a fascicle. So what I find interesting is that the fascicle looks a little bit like the whole muscle. And so here's the perimysium again, right there. But then check this out. Each fascicle is further broken up into little compartments. And each of these compartments right here is indeed a muscle cell. So each one of these is a muscle cell. I know it doesn't look like it right out of the gate. I'm not sure what you're thinking. But this tissue right in here that surrounds each of the muscle cells within the fascicle is endomysium. So many layers of connective tissue for support. So if I were to pull out, again, sort of, reaching within the fascicle and pulling out one of these things, this right here is a muscle cell. Now, it's called a muscle fiber or a myofiber. So myo, the prefix for this means muscle. So myofiber also means muscle cell. That can be kind of confusing to some students. So this is a muscle cell right here. This whole thing is a muscle cell. And what's interesting is the membrane of a muscle cell, because every cell has a membrane. Every cell, muscle cell, the membrane, we're going to call it sarcolemma. And sarca is a prefix that means flesh. So it comes up a lot when you're discussing things within the muscular system. So sarcolemma, sorry for all the terms right out of the gate, but it's helpful. Sarcolemma is the membrane that surrounds the muscle cell. And within the muscle cell, of course, you have a nucleus. What's interesting about skeletal muscle is that it's multinucleated. And there's going to be a better picture coming that shows that. So there aren't a lot of cells in the body that have more than one nucleus, and the reason is the incredible length of a muscle cell. And I think the reason that it's called muscle fiber instead of it just being muscle cell is that it's really long. And so it's longer than it is wide. So it's like a fiber. The muscle cell is. And the reason for the multinuclei is that it needs to make so much protein, and the distance is so vast within the muscle cell that it's more efficient to have more than one genetic code doing that, because as you know, the DNA is the code for protein. So within one muscle cell, there's all kinds of, check this out, there's all kinds of sheets of protein, which sort of look like little salamis right here. All these cables of protein are called myofibrils. Now, don't confuse that with a myofiber. A myofiber is the whole cell. So this is the whole cell right there. So each one of these is a myofiber. And the myofiber is composed of many different kinds of proteins, but these proteins can be referred to as just simply filaments. There's thick filaments and thin filaments. So we're going to come down to this level because it's the sliding of the thick and thin filaments past each other that's going to make the whole muscle contract. So pretty cool, I think. It takes a little while to get comfortable with this hierarchy. And again, this yellow membrane within the cell is our good old smooth endoplasmic reticulum. But in the muscle fiber, muscle cell, we call it the sarcoplasmic reticulum because it's the endoplasmic reticulum within the muscle cell, sarcoplasmic reticulum. That's going to be a storage area for calcium, which is important in order to initiate muscle contraction. And so ultimately, let's get down to it. We have these thick proteins and these thin proteins. Let me go back here. So the thick one here in red, and then here's this thin one in blue. The thick protein is referred to as myosin. Okay, so it's a huge protein, and what's significant about it is that it has these sort of cross appendages that come off of it. And these these cross bridges that come across it are sort of like the head of a golf club. I like to think of it as it. And then the thin filament is made up of, each one of these are proteins, so this is a quaternary level of a protein, but this whole thing collectively is called actin. So these are little actin subunits right here, and there happens to be 2 chains, actin filaments. And so what I'm getting at is that the myosin actually interacts with the actin, and it attaches onto it. So like an arm, it reaches up and it grabs the thin filament. And where it grabs, it's highlighted here in blue, is called the active site. It's the place where the actin attaches to the myosin. And so what we're going to see is that there's these subunits called sarcomeres within the muscle fiber. And I'll come back to this. These units, you see this line right there and this line, these sarcomeres, which is what these units are, and I'll come back to that, are going to shorten. And so that's what the sarcomere is composed of. And mere means part. Sarca again means flesh. So there's these fleshy parts that are repeating within the muscle cell that are going to be shortening. And so here's a close-up of that. And so ultimately, I mentioned this adenosine triphosphate as being a very important energy molecule. And so this thing is produced within the cell, and you may know this somewhere from your biology past, but it's produced within an organelle called the mitochondria. And so that's where it's coming from. Now food is going to be going, I won't get into this conversation, though I could. The food goes in, it's broken down, and then this chemical adenosine triphosphate comes out. And this is going to interact with the myosin protein. Okay? And so the myosin, as it turns out, check it out, it has many of these cross bridges that are coming out right here. So this is what we mean by one sarcomere. This is within a muscle cell. And so this is on the molecular level. Do you see how myosin is really thick? And then these blue lines are the actin thin filaments. Okay, so when scientists were first looking at this under the microscope, they began to describe it with various letters, because there's sort of zones. There's a light color zone because this area right in here is only actin, and so it appears under the microscope as kind of lighter. And then do you notice here how this is sort of thin, thick, thin, thick, thin, thick? So this is kind of a darker area right in here, dark area. So this area in general, this dark area, when you look at this under the microscope, is referred to as the A band, this dark area. And then this lighter area is an I band. So it's light, dark, and then this would be the I band over here. And so what we say is, when you again look at this under the microscope, these proteins come down, it sort of looks like a zigzag right in here. And so this is referred to as the Z line, the letter Z, Z line, and here's another Z line. Okay, so this is what we mean by the definition of a sarcomere is from Z line to Z line. So that's one functional unit. And what's going to happen is the myosin is going to grab onto the actin filaments and pull it closer together. And so what's going to happen is the H zone, which is in the area here, is going to decrease in size as the Z lines come closer and closer together. Okay, so this is what we're talking about. And you're like, well, why would it contract? You know, what's going on? So again, more pictures to sort of understand this. This is one muscle cell. A muscle cell is also called a myofiber. Within the myofiber or muscle cell, there's a nucleus, there's all kinds of mitochondria. The mitochondria make ATP. And this yellow is a whole bunch of membrane within the cell called sarcoplasmic reticulum. We know it as just simply endoplasmic reticulum. And it surrounds all of these sort of big cables of protein. So the muscle cell is kind of like, again, sorry for so many analogies, it's kind of like a bag of spaghetti. It has all these like strands within it. And all these proteins within it, right here, here's one being pulled out, are myofibers. Myofibers. Okay, and so the myofibers, or myofibril, I'm sorry for misspeaking that, the myofibrils are composed of proteins. The myofibrils are the proteins within the myofiber. And so you could see here, here's one Z line, and here's another Z line right here. So this is what we mean by one sarcomere. And then as you can see here, though it's not shown, this is the A band, which is dark. It's dark because it's superimposing the thin and thick filaments. And here's the I band, which is a little bit light. Now under the transmission electron microscope, very powerful, you can see light, dark, light, dark. But under the light microscope, you just simply see lines that we like to call striations. It's one of the characteristics of skeletal muscle is that it's striated. And so you can actually see that under the light microscope. It's pretty cool. And so what's going to happen is the thick filaments, which are called myosin, are going to be grabbing along the thin filaments and pulling it in like a clothesline. And the sarcomere is going to get short. And that's basically what's happening in muscle contraction. And so again, breaking the muscle down to its sarcomere subunits. Again, here's an actual picture of a transmission electron microscope. You can see I band, A band, I band. So here is ultimately the Z line right in here, which is what that is. This dark area right here is the Z line. So that's sarcomere from there to there. And then this is dark because it's a combination of light, thin and thick filaments right in here. And so here's a close-up, again, transmission electron micrograph of a mitochondria. The mitochondria makes a tremendous amount of ATP, and again, food is burned in the mitochondria, and it's ultimately used, the energy of burning food, the oxidation of food, is to attach the third phosphate. The phosphate is a negative functional group, and it attaches that to ADP, and ADP doesn't really want it because those phosphates are negatively charged, so it takes a tremendous amount of energy to do it. So that's the reason why we are eating food, is to provide that, and also source of atoms as well. But the phosphate attaches onto it, and you make ATP, and look at this, millions of it are consumed per second, and so it's kind of staggering. And so again, here's a picture of, you might recognize this. This is ATP, and what's going to ultimately happen is, picture this as being the myosin. Okay, here's the myosin, and here's one of the heads sticking out, and you're like, what is all this? These are amino acids which compose the protein of myosin. What's going to happen is ATP is going to donate one of its phosphates. And so let me bring it over like this, and it's going to stick it onto, this is negatively charged. And when something's negatively charged and it interacts with a protein, it's going to cause a conformational change to occur, which ultimately means that it's going to change shape, it's going to move. And so when it moves, it's going to be, this is what we mean by like being able to do something. So the myosin's going to move, and as a result of ATP donating one of its phosphates, it becomes ADP. Okay, so what we call this, when water splits a phosphate off, we call this the hydrolysis. Okay, that's kind of a generic term, meaning like the splitting of a molecule with water. Okay, so here's where it comes down to it. This is how the muscle's ultimately contracting. It's all about the interaction between the myosin and the actin. And it's called the sliding filament model, because that's actually what's happening, is the thin filaments are sliding closer together. And the reason the thin filaments, which are actin, the reason the thin filaments are sliding close together is that myosin is pulling it in. Myosin's pulling it in. And so check it out. What happens is, you could look at this cycle and start it in any number of ways. It's kind of hard to understand it in a still image like this, but ultimately it's a cycle because it repeats over and over again. Let me see if I can enlarge this so you can see it. So check this out. What we're going to have happen is the myosin head is going to be released from the actin, because ATP is going to give up one of its phosphates and it's going to stick to it, and that's going to cause the myosin head to let go of actin. And when it lets go of one of the active sites, it then is enabled to attach to a new active site and then pull it closer. And as it pulls it inward, that movement in is going to dislodge the phosphate. So as it grabs a hold of the actin and pulls it, the phosphate falls off and then it requires another phosphate to attach in order to disengage it. And so I understand that that's a little tricky to understand. So let's take a look at this concept a little bit more and closely. Check this out. So ATP ultimately is causing the myosin cross-bridge to let go, let go, let go, let go. But what's moving it is the attachment, okay? And so let's go over here to this one. I find that this is pretty helpful. So here's the myosin, and here it is attached to the active site. Here's the myosin head, and so the ATP is needed to get myosin to let go of actin. And so as it turns out, these myofilaments are in contact with one another, and so the myosin attaches to the actin, and it's pulling, and it's going to pull it inward. See it pulling in? So in order for it to let go, a phosphate needs to be attached to the myosin cross bridge. And so when ATP attaches to it, it lets go, and then it allows it to engage with the next active site. So there it is, grabbing it, and then pulling it. And as it moves it, it doesn't show this in the detail, but the phosphate falls off. And then another phosphate comes on, and then it pulls it, and the phosphate comes off. Another phosphate comes on to let go. It pulls it in, and the phosphate comes off, and then another phosphate comes on to let go. So it's the phosphate causes it to let go. And then when the myosin head pulls it in, that causes the phosphate to come off. And so it's kind of neat that way. I'm not sure if that was crystal clear, but it's potentially clear enough for us to continue our conversation, because that's what we're going to do. So it's a little bit of a cycle. And so basically what's happening is the thin filaments are coming toward the center, and so the H zone, if you recall that, is disappearing inside. Okay, so how do we regulate? So this is a related topic, but it's another concept. How does the muscle regulate protein contraction? Because if you're following this, it just means that if there's ATP in a muscle cell, that means that that's going to provide the energy necessary for muscles to contract all the time. There must be some way in which muscles regulate their contraction. So in other words, when you don't want a muscle to contract, something must be stopping it. So you might have guessed that it probably has something to do with the myosin head interacting with the actin. As it turns out, that these actin active sites are at a resting state blocked. And so the actin, let me repeat that, the actin active sites are blocked. And so the myosin head can't attach to it. So you can say it's the actin binding sites. Here it says the myosin binding sites. It's fine. But they're blocked. They're blocked by a filament type of protein called tropomyosin. So tropomyosin at rest, so this is like blocking the muscle from contracting. The tropomyosin, this filamentous protein right here that coils around the thin filament, is blocking the cross bridge from attaching. Okay, so how do you release it if you want the muscle to contract? Well, there's yet another protein. It's kind of neat, don't worry. There's another protein shown here. It's got 3 small subunits in purple, and it's called troponin. So troponin is a protein that sticks to tropomyosin. You see that? So here's troponin, and it's sticking to tropomyosin. When the muscle cell wants to contract, over here, let me illustrate this, more pictures to come later. Over here in the sarcoplasmic reticulum within the muscle cell is a storage of calcium ions. Yeah, so calcium is not just stored in bone, it's also stored in muscle inside the sarcoplasmic reticulum. And so you ready for this? Calcium is released from the sarcoplasmic reticulum. It's released through a calcium ion channel. More to come on that later. So much to talk about, trying to keep it short. So calcium is released, and as you can see it here is in these green balls. When calcium is released from the sarcoplasmic reticulum, calcium also has a charge to it. It's a +2 ion, just like a phosphate was a -2 charge. When charged substrates interact with proteins, they cause them to change shape. And so check this out. Calcium binds to troponin. And so when calcium binds to troponin, it changes the shape of troponin. Changes the shape, delta means change, changes the shape of troponin, which then changes the shape of tropomyosin. And that causes, check it out, it causes the tropomyosin to move slightly. And when it moves ever so slightly, it exposes the binding site. Oh, now that the binding site is, do you see here's a close-up. Now that the binding site sort of look like olive pits right here. Now that the binding site is available, the myosin cross bridge can grab a hold of it and pull it in now that it's available. So this sort of troponin-tropomyosin complex regulates muscle contraction with the help of calcium. And so how do you know to release calcium? I won't get into the detail of this, but I'll just simply state it. If you want your muscle to contract, a neuron sends a nerve impulse, which is referred to as an action potential. And the nerve, which is here in the axon terminal, doesn't actually touch the nerve cell, but it comes very close. And where it comes close is called the synapse. We call that the neuromuscular junction. And a chemical neurotransmitter comes across and ultimately allows the calcium that's being stored in the sarcoplasmic reticulum to be released. So the nerve impulse causes the calcium to be released. And if the calcium is released, remind you, when the calcium is released from the sarcoplasmic reticulum, that's going to interact with the troponin, which moves the tropomyosin, which allows the myosin, which is the thick filaments, to interact with their cross bridges with actin and pull the actin filaments closer together, which shortens the sarcomere. And so overall, the entire muscle fiber, which is muscle cell, gets short. And if the muscle fiber gets short, then all of the muscle fibers within the fascicle get short, and then all those fascicles which are inside the muscle, and so therefore the whole muscle contracts. This is a cool picture of showing that nerve impulse here. This is, each one of these is a muscle cell or a muscle fiber. Notice the striations here. Again, you can see this with the light microscope. Let's review. This was the A band. Do you remember the dark was the A band and then the light was the I band? So it's A band, I band, A band, I band, A band, I band. Okay. And so within the I band is the Z lines. And so that's the sarcomere. Okay. So if these are each muscle cell, this would be surrounded by, you might know this, surrounded by endomysium. Endomysium. That's surrounded. Here's the multinucleated cells. And so here's a nerve cell that's telling the sarcoplasmic reticulum to release calcium. It's releasing calcium. And so when it releases calcium, that's going to cause, look at this. Here's, do you see this part right here, the axon terminal? That's right over here, the axon terminal. It branches so there's more surface area. It doesn't actually touch, physically touch. It comes very close. So this is what we call the neuromuscular junction, where they come together. So we'll talk about this in a little bit later video, but the neurotransmitter comes across from these vesicles and it interacts with these proteins right in here. This is acetylcholine. Interacts with this, which causes a charge differential to occur, and calcium is released from the sarcoplasmic reticulum. It interacts with the troponin, which causes the tropomyosin to move away from the binding site. Then myosin just simply, hey, I can grab it and pull it. And when it grabs it and pulls it, a phosphate falls off, and then a new phosphate is added, and it causes the myosin to release from the actin filament. So it requires energy to relax and let go. And so in conclusion here, check this out. You could see basically what's happening when the sarcomere contracts, the Z lines come closer together. And notice that it doesn't show it here, but this is the H band. The H band disappears, and the A band gets smaller. Do you see that right there? So why is this happening? Because this filament is being pulled in, pulled in, pulled in, pulled in, and this one's being pulled in. So it's the thin filaments that are sliding past the thick filaments. The thin ones are sliding past the thick ones. And again, I'm cognizant of the fact that this is a little abstract and hard to follow, but these are actual electron micrographs showing the fact that here's a relaxed state right over here. Do you see that? Here's the Z lines far apart from one another, and here they are coming closer together. The Z lines are coming closer together. And so the reason this area is dark, the A band is considered to be dark, because it's overlapping thick and thin. This is the I band over here that's light, which is over here. It's the light, the I band, and then the Z is over here. So sarcomeres are getting shorter. So let me conclude with this. Sorry. Let me conclude with this is, I find, a pretty useful summary of what we're just talking about. You can see, here's the thick filaments shown here in purple with the myosin. Here's the thin ones in green. And what's happening is when the muscle contracts, look at those little heads pulling and pulling. So that requires a lot of ATP to let go. It pulls them in, and then notice the H band totally disappears. Okay? And so the sarcomere shortens. And as the sarcomere is shortening, the whole muscle cell, it gets short. And so the I band also narrows as a result of this. So believe it or not, a brief look at muscle contraction. I think it's kind of neat. It requires some time to think about it, but it's really fascinating how all these proteins work together, and ATP is involved and calcium is involved. And so ultimately it's a very cool thing how proteins interact with one another and they change shape and they slide past each other with the help of phosphates and calcium. So I hope you enjoyed this video on muscle contraction. Thanks for watching.